We're going to talk about Gregor Mendel, his experiments, and Punnett squares. These are our learning goals for today. We're going to describe the experiments that Gregor Mendel did with peas. We're going to predict the inheritance of simple traits based on the laws of probability. We're going to determine the possible gametes of a breeding. And we're going to construct a Punnett square to determine the genotype and the phenotype of the offspring from a monohybrid cross. Gregor Mendel was an Austrian monk. He lived a long time ago. He was a mathematician, not a biologist, and he used math to figure out how traits were passed from generation to generation. We call him the father of genetics. Mendel used garden peas for several reasons. One is they're easy to grow. They grow quickly, so he was able to observe many generations in a short amount of time. And they were easy for Mendel to control which flowers from which plants were able to pollinate. In other words, which plants were able to breed with each other. So here is Mendel's experiments. He took pea plants that produce purple flowers, always purple flowers and no other color, and he mated those with pea plants that produce white flowers, only white flowers, and no other color flowers. This is called the pea generation or parent generation. And what he found was that their offspring were all purple flowers. Not one white flower, not any other color, but they were all purple. We call this the F1 generation or first family generation. Then Mendel took plants from this first family generation, these all purple flowers, and he mated them with each other. And what he found was that their offspring always produced purple flowers and white flowers, but there were always 3 purple flowers for every white flower. They were always in the ratio of 3 to 1 in this second family generation. Now Mendel repeated these experiments on other traits of pea plants, from seed color, seed shape, height of plants, all different types of characteristics, and he always got this 3 to 1 ratio. And because he got that 3 to 1 ratio, he was able to work out how traits were transmitted. And we're going to watch that and observe that when we do Punnett squares. Punnett squares allow us to predict the offspring of a mating between any 2 individuals. In this case, we're going to predict the offspring of a mating involving one trait, and that's flower color. Suppose we have 2 flowers, and the alleles for flower color are big R, capital R, for red flower color. Remember, capital means dominant. And little r for white flower color. Remember, lowercase letters mean recessive. So the genotype of our 2 parents is heterozygous. We have 1 dominant gene and 1 recessive gene in each parent. So they're heterozygous. Now, remember that these letters stand for sections of a chromosome, genes. So let's look at some pictures to help us visualize that. What we're really talking about is these flowers have a pair of homologous chromosomes, and on these chromosomes is a section of DNA that produces a protein that controls the flower color trait. So here are 2 homologous chromosomes in one parent, and here are 2 homologous chromosomes in another parent. Each chromosome carries 1 version or allele of the gene, Now, we know in meiosis that these two homologous chromosomes are going to become separated. And if we imagine the individual on the left to be a male, then when this individual creates sperm, there could be one sperm with a capital R, and there could be one sperm with a little r. There could be one sperm that receives the homologous chromosome with the dominant gene, and there could be a sperm form that receives the homologous chromosome with the recessive gene. Same thing for the female. She could produce an egg that contained the chromosome that carried the dominant version of the gene, or she could produce an egg that carried the chromosome with the recessive version of the gene. So these are all the possible gametes, sperm and eggs, that these two plants can produce. We can use a Punnett square to predict all the possible offspring that these sperm and egg can produce. Now, Punnett square is right here. Notice it's divided into 4 sections. We're going to put the sperm across the top and the eggs down the side. Notice we have one sperm above each column, and we have one egg next to each row. Now we're going to fill in by moving the sperm down and filling in the column. So we can put one sperm there with a dominant version of the gene, another sperm there with a dominant version of the gene. We can put a sperm there with a recessive version of the gene, and another sperm there with a recessive version of the gene. Then we can bring our egg across with the dominant version of the gene and put one in each one of those squares. And then we can take our egg with the recessive version of the gene and put one in each one of the squares. Now we've put sperm and egg in every possible combination in the Punnett square. And the thing we have to remember is that the sperm and egg represent possible zygotes. Sperm and the egg fertilized and produce zygotes, or fertilized eggs, which would become possible new plants. So these are all the possible offspring that can be produced by the two individuals we started with. Now, let's look at the different genotypes and phenotypes of the offspring. So let's organize them. Okay, now we've organized the possible offspring by their genotypes. Here we have one egg that has 2 copies of the dominant version of the gene. This is homozygous and it's dominant. Here we have 2 possible offspring. They're heterozygous. They both have 1 copy each of the different versions of the gene. We have 1 copy of the dominant gene, 1 copy of the recessive gene. And we have 1 offspring that's homozygous and has 2 copies of the recessive gene. So what are our possible phenotypes? What will these individuals look like? This individual would be a red flower. These 2 individuals would both produce red flowers, and the last one would produce white flowers. So we have our genotypes on the left and our phenotypes on the right. And notice our offspring are in the ratio of 3 dominant traits to 1 recessive trait, just like Mendell got in his experiments. Now we don't have to always draw sperm and eggs and zygotes. We can just use the different versions of the gene, the different alleles arranged across the top and down the side of the square. We can fill in the squares. And then we can look and see what the genotypes and phenotypes of the possible offsprings would be. So in this case, we get the same thing as before. We have one genotype that's homozygous and dominant. Its phenotype would be a red flower. We have two individuals that have a heterozygous genotype. They have one of each allele, one of each version of the gene, one dominant, one recessive. Phenotype would be red flowers. And our last one is homozygous recessive. It has two copies of the recessive version of the gene, two recessive alleles. And its phenotype would be a white flower. Now, when we're talking about Punnett squares, and you guys will be working problems, there's some terms that you should know and remember. If an individual is purebred or true breeding, that means they're homozygous. They could be homozygous dominant or homozygous recessive. Right? They could have two copies of the dominant gene or two copies of the recessive gene. If we say an individual is a hybrid, that means they're heterozygous. That means they have two versions of the gene. They have two alleles. They have one dominant and one recessive.